Hello and welcome to our podcast. I'm Justin, and today we're going to explore some amazing science that helps to keep our planet healthy. Every day, human action like releasing chemicals, dumping waste, and cutting down forests can harm the environment. For example, pollution and climate change are causing species to disappear and ecosystem breakdown. Typically, you might think we only need a few water samples or a quick fish count to gauge a habitat health. Yet, these methods can miss the very first signs of stress. It's like waiting for someone to collapse before taking their pulse. What if we could spot trouble at cellular level long before the entire population starts to die off? Every year, our river and soils absorb millions of plastic, chemicals, and waste. Microbes are the first responders to this assault. But until now, we have lacked the tools to eavesdrop on their early distress calls. What we really need is a molecular alarm system that rings the moment an ecosystem begins to falter. That's where cutting edge techniques called functional genomics and proteomics come in. Think of genomics as reading all the DNA in a sample of soil and water. That tells us which organisms are present and what they might be capable of doing. Proteomics, on the other hand, is about looking at all the proteins those organisms make. It's like checking the machines and workers that cells are using right now. Just like a car dashboard has warning lights to alert you to engine trouble. This method look for molecular signals of stress and change in an ecosystem.
That sounds almost like science fiction, but it's real science.
It really is. And today, I'm joined by my friend and colleagues Jazuli and Badrul to break down these topics. Jazuli will dive into how genomic tools help us understand ecosystems, and Badrul will explain what proteomics reveals. Finally, we will tie it all together by discussing how these tools help us clean up pollutions and conserve nature. Let's dive in. So, Jazuli, tell us what exactly is genomics and how can it help us monitor nature?
Thanks, Justin. Let's start with genomics. Imagine you scoop up a handful of river water or a pinch of forest soil. What if I told you that this single sample could contain the DNA of thousands of organisms? Traditionally, we have to examine organisms under a microscope or grow them in a lab one by one. But with metagenomics, we can extract DNA directly from the sample and sequence it all at once. It's like mixing together every instruction manual from the living things in the sample and reading them at high speed. We find out instantly here are the bacteria present, including the plants, fungi, and even viruses in the mix, along with all the genes they carry.
That's fascinating. So, is this technology already being used around the world?
Absolutely. For example, scientists have used metagenomics to compare soils from very different places, like frozen tundra versus tropical rainforest. Each environment had its own unique microbial community and genetic toolkit. In studies of lakes, researchers collected water from high mountain lakes and warm lowland ponds. They saw certain groups of bacteria, like cyanobacteria or proteobacteria, dominate different lakes, and the genes they carried matched the lake's conditions. Some lake bacteria had genes for dealing with cold, other genes for coping with salt and intense sunlight.
Wow. So by reading these genes, we can tell what functions microbes are performing. But what about looking for specific processes like nutrient cycling?
Great question. Many microbes have genes for nutrient cycling, like capturing nitrogen from the air or processing sulfur compounds. By counting those genes, we learn how a community keeps nutrients in balance. For example, finding genes for nitrogen fixation in a forest soil sample tell us those microbes help plants get the nitrogen they need. If pollution disturbs the balance, changes in those genes could be an early warning that the ecosystem is stressed.
And we have also heard about the term eDNA thrown around. What is that all about?
Environmental DNA, or eDNA, is another powerful tool. All organisms leave behind DNA traces in their surroundings. For example, fish shed skin cells into water, plants release pollen and leaves in forests. Even animals leave hair, feathers, or saliva. Scientists can take a water or soil sample and find DNA fragments from all sorts of creatures. It's like nature's own surveillance system. For example, by sampling river water, researchers have detected every species of fish and amphibians present, even rare or shy ones, without ever catching or seeing them.
That's incredible. And I've even heard about airborne eDNA. Is that a real thing?
It is. New techniques let us use air filters in forests or cities to capture genetic material floating around in dust and pollen. This airborne eDNA has picked up DNA from insects, plants, and even small mammals. It's like nature is constantly broadcasting information, and we're just tuning in.
So, to sum up, genomics and eDNA let us answer two big questions: who's here and what can they do? But once we know that, how do we use this information to actually clean up pollution?
Definitely, once we know which organisms and genes are present at the site, we can make smarter cleanup plans. Okay, you imagine a lake contaminated with oil. We take a sample and sequence the DNA. If we find lots of oil-eating bacteria and the specific genes they use, like genes for oxygenase enzymes, that's a green light. We might then add a little extra fertilizer to feed those bacteria, or even introduce more of them if they are scarce. These strategies have names. Adding nutrients is called biostimulation, and adding microbes is called bioaugmentation.
So, genomics is not just monitoring, it's a guidebook. Here is the local experts. Use them. I love that. Thank you, Jazuli, for that eye-opening sharing. Now, Badrul, could you tell us more about proteomics and how it complements genomics?
Thanks, Justin and Jazuli. Now, let's switch gears to proteomics. If genomics is reading the genetic blueprint, proteomics is about examining the actual machinery built from that blueprint. Proteins are like the machines and workers in a cell, they carry out the work. Proteomics looks at which proteins are present and how abundant they are. This tells us what is really happening inside the organism at that moment. Think of protein as a cell's workforce. When pollutants hit an organism, like heavy metals or microplastics, and its proteins change dramatically. Proteomics will detect this shift, like which stress protein surge or which detox enzymes was activated. This gives us a real-time molecular snapshot of pollution's impact long before fish die or ecosystem collapse. It's an early warning system nature never had before.
Proteomics is like tuning into an orchestra of molecules. Very poetic. But why do scientists sometimes focus on muscles?
Great question. Muscles are filter feeders that accumulate pollutants, acting as bioindicators. One iconic example is the Mussel Watch program, which was run by National Centers for Coastal Ocean Science (NCCOS) in the US and Europe. Mussels in the Mussel Watch program show altered expression of stress response proteins such as superoxide dismutase, glutathione, or transferase when they are exposed to oil, heavy metals, or toxins, serving as early warning indicators. So here is how it works. Basically, the muscle filters seawater, concentrating pollutants in their tissue. And then scientists will use proteomics to analyze their protein, specifically from gills and blood. When exposed to heavy metals like cadmium, muscle overproduce stress proteins like catalase and superoxide dismutase. So using proteomics, we can detect this protein spike which act as a red flag for pollution. In a key study by Campos in 2015, proteomics revealed how microplastic and chemicals triggered cellular damage in muscles, disrupting energy metabolism and immune responses. This is not just lab work. By comparing protein profiles across sites, we map pollution gradient in real time. Proteomics literally turns muscles into living pollution sensors.
Fascinating. But does this approach extend to microbes as well?
Absolutely. Take the bacterium Pseudomonas putida, which was famous for eating oil. By studying its proteome, scientists identified which enzyme it used, like dioxygenases, which are chemical scissors that chop up oil. Seeing those enzymes in action tells us the bacteria are actively cleaning up a spill down toxin. So by tracking protein expression under different conditions, we can optimize cleanup by playing with the parameters, such as adjusting pH, temperature, or nutrient to boost degradation. We even spot stress protein signaling when the bacteria struggle and letting us tweak strategies. In one breakthrough based on a study from Yi Hui in 2011, proteomics, if combined with genomics, reveal P. putida full pollutant digesting toolkit. This paved the way for designing supermicrobes via synthetic biology to tackle stubborn contaminants like PCBs or pesticides.
So, if genomics tell us who's here and proteomics tell us what they are doing, it's like a real-time report card on our cleanup crew.
Precisely, and if we see mostly stress response protein instead of cleanup enzyme, we know conditions need adjusting, maybe more oxygen or nutrients.
So, let us tie it all together. How do we use genomics and proteomics in real-world cleanups? Well, first, genomics help us to identify the right players. If we sample a polluted site and find microbes with genes for breaking down oil or heavy metals, we know who to harness. Then, proteomics confirms they are active and tell us if they need a boost.
It's like tending to a garden of helpful microbes, giving them exactly what they need to grow strong.
Exactly. In an uranium contaminated groundwater project, researchers discovered that geobacter could precipitate uranium out of solution. Genomic and proteomic tests confirm the microbe's activity, and by feeding it with the right nutrients, they lock up the uranium safely. And during oil spill cleanups, scientists monitor the sites over time. Genomic data shows that oil-eating bacteria increasing in number, while proteomic analysis shows their enzymes kicking into action. As the oil is break down, stress-related protein fall and normal protein returns, signaling recovery. That wraps up our journey through genomic and proteomic in ecosystem health. To summarize, metagenomic fingerprints point us to the organism that can help clean up or maintain an ecosystem, and proteomic analysis confirm the work is happening, and show how things evolve over time. We have seen microbes cleaning up toxic waste, monitoring oil spill recovery, and more feats that are impossible just a decade ago. Looking ahead, imagine real-time sensors that sequence DNA and protein on the spot, alerts that flash when something dangerous appears, or even designer microbes crafted by synthetic biology to gobble up plastics. The field is moving fast. The key takeaway is this: genes and proteins are the hidden language of life, and now we have the tools to read that language in the wild. By listening at the molecular level and combining genomics, proteomics, metabolomics, transcriptomics, and AI, scientists get an ever clearer picture. We can intervene earlier, make smarter decisions, and truly restore the health of our planet.
If you enjoyed this episode, please share it with anyone curious about science and the environment.
Thank you again for listening, and take care of our planet and each other.
And that's a wrap from us. Keep exploring and keep our planet healthy.
Goodbye, everyone, and stay curious.